Hello and welcome back to Viewing of Prepared Slides Part 2, and today we are going to be focusing on oil immersions. Now, oil is generally used with high-powered lenses like 100 times magnification. To know for sure that that objective lens is designed to be used with oil, there are usually some type of special markings. For example, it could have a gray or silver band going around it, indicating that that objective lens is used with oil. But before we get to oil immersions, we have to get through the other objective lenses first. Before we begin, we have to pick out a slide. Today I will be using the bacteria slide. For this demonstration, I will be using acid-fasting stains, or mix. Acid-fasting stain is able to differentiate between two types of gram-positive cells, those that have waxy mycolytic acids in their cell walls and those that do not. There are two different methods for acid-fasting staining. They are the Ziehl-Neelsen technique and the Quignon technique. Both of these techniques use carbon fusion as the primary stain. The waxy acid-fast cells retain the carbon fusion even after decolorizing agents, like an acid-alcohol solution, is applied. The final step of acid fasting is to apply a counterstain. Usually it's methylene blue. This counterstain renders non-acid fast cells blue, while the acid fasting cells remain red because they still have all of these carbon fusions. To begin the experiment, smear the specimen directly onto the slide. Then let it air dry. Once the specimen has air dried, you can now move on to the next step. Using the Ziehl-Neelsen method, use heat to infuse the carbon fusion into the acid-fasting cells. You can use a Bunsen burner for this. Once this is done, you can now begin staining. After you are done staining, you can now place your slide under the microscope. When placing the slide on the microscope stage, you want to move the mechanical stage or the stage clips apart so that the slide slips in between the stage clips neatly. Using the revolving nose piece, rotate to the scanning or 4 times objective lens. While you're on scanning, adjust your slide holder. Use the coarse adjustment knob to move the stage up closer to the objective lens. Doing this brings the specimen into focus. Then use your fine adjustment knob for your fine tuning. This knob is used to improve the clarity of your image. If needed, use the iris diaphragm to adjust the brightness of the image. Once you've looked at your image while in scanning mode, you can now move up to low power. Repeat the same steps that you did for scanning. Use the fine adjustment knob to adjust the focus of your image, and use the iris diaphragm as you see fit. After observing the specimen through low power and the image is in focus, you can now switch to high power. Repeat the same steps that you did for low power. When on high power, make sure not to use the coarse adjustment knob. This knob is only used for low power. After observing this biological sample through high power, you can now move on to the immersion oil lens or 100 times magnification. When switching to immersion oil lens, make sure to turn the objectives halfway in between. Then add a drop of immersion oil to the slide, preferably in the middle of the slide where the specimen is. Once you've done that, you can rotate the immersion oil objective into place. Remember, once the oil has been added to a slide, the objective lens should not be switched back to an objective lens that does not use oil. This could cause problems for that non-oil objective lens. You can adjust your slide to get a better focus. Using adjustment knobs, you can move your slide forward or backwards or right to left for a better image. When you're finished with the microscope, use the coarse adjustment knob to bring the stage all the way back down. Rotate the objective lenses halfway so that they are out of the way. Then, remove the slide. To clean off the immersion oil lens, only use lens tissue paper. Any other type of paper could scratch the objective lens. You also never want to touch the lenses with your fingers. Oils from your hands can smudge the lens and could potentially scratch the lenses. When you've cleaned off your microscope, make sure to rotate the nosepiece back to the lowest power setting, lower the stage completely, wrap the power cord up neatly, put the dust cover over the microscope, and return the microscope to the cabinet. Remember, while carrying the microscope, grip the arm of the microscope with one hand and place the other hand underneath the base for proper support.